So this is binary fission, which is a type of cell division that occurs in prokaryotes only. And so here the DNA is attached to the cell membrane, and then it replicates as the cell expands, and then the cell eventually undergoes cytokinesis, which results in 2 daughter cells. And this is a form of asexual reproduction, and thus there is no genetic variation. This is a quick overview of interphase. So it's G1 phase to S phase to G2 phase. And so here is G phase, so it's just growing, and then S phase is when the DNA and all of the chromatin there replicates. And so G2 is growth and preparing to divide. And so here is what happens after G2, which would be the beginning of mitosis, which is prophase, where sister chromatids form at centromeres, and so that by way of the replicated DNA. This is G0, which can occur after cytokinesis, which is when a cell is essentially just at rest. And so an example of this type of cell would be a neuron. Here's a quick overview of the cell cycle in just a diagram format. So G0 or G1, and then if G1, then to S, then to G2, then to mitosis, then to cytokinesis, and then the cycle repeats. And then the red sticky notes represent cyclins, which regulate the cell cycle at different checkpoints, and the levels of cyclin are the highest at mitosis, as seen here.
Now let's go more in depth in the process responsible for growth and development, mitosis. As aforementioned, mitosis is the aspect of the cell cycle that leads to cellular division, where the conclusion is the replica of 2 daughter cells. The first stage involves chromosomes condensing in the familiar X shape, creating 2 sister chromatids, and the centrosomes, containing centrioles, radiate microtubules and migrate to opposite sides of the cell. This stage is called prophase. Next, the nuclear envelope that contained the chromosomes break down, and the microtubules from the centrosomes attach to the chromosomes. This stage is called prometaphase. After prometaphase, all the chromosomes align with the center of the cell, making up the stage called metaphase. This leads to anaphase. In anaphase, the sister chromatids that make up each chromosome are split apart and travel to opposite sides of the cell to prepare for telophase and cytokinesis. In cytokinesis, along with telophase, the cell involved in mitosis is completely separated into 2 daughter cells. The nuclear envelopes of the 2 cells reform and the chromosomes decondense. Next, cytokinesis causes a cleavage furrow, where the cells pinch off their membranes and completely separate. From here, one of the cells continues back into interphase to continue the cycle, while another will leave the cycle to enter the aforementioned G0 phase.